Well, hello there, Biology 30s. Today we are talking about the Hardy-Weinberg equilibrium. So you'll need your population genetics notes guide. And Hardy-Weinberg is this mathematical model. So kind of like how a map of the Earth, it's a model, right? It's not perfect, but it gives us a way to make predictions about things. And so we can actually predict the frequencies of alleles in a population. And if the population is stable, the allele frequencies or the number of individuals or the percentage in a population with a particular allele is not changing. They're in equilibrium. So your Hardy-Weinberg, the genotype equation is given to you in your data booklet. And we're going to talk quite a bit about this one. And then there's another equation. I would honestly recommend that you memorize this one. And again, we'll talk about what each of the terms in this equation represents. So, one of the very first things that you may see on a formative or a summative or on the diploma is the conditions or the assumptions, the criteria in order for the Hardy-Weinberg equilibrium or principle to be true. So the first one is that there needs to be a large population. So if genetic drift is occurring, it's happening in small populations. And we don't want that. We want there to be no chance or random events. And so we don't want the genetic drift to be occurring. And sorry, I've got a little bit of a punctuation problem here. There needs to be a large population, not a small one, because otherwise, any changes in population size can lead to a founder effect or a bottleneck effect. Okay, so that's the first thing that we need for a condition or criteria or assumption for Hardy-Weinberg. Like I said, this could be a numerical, it could be a multiple choice question, but you're probably going to see these conditions or criteria somewhere on a formative or summative. The second assumption, in order for the Hardy-Weinberg equilibrium equation to be true, is that there's no migration, there's no gene flow. So individuals are not immigrating, they're not coming into a population, because if that was true, you'd be adding alleles into a population. There cannot be any emigration. Why? Because that would mean that you're removing alleles from a population. So no migration. Okay. Third condition, no net mutations. So we talked about like Benji was this purple, blue-skinned person, and the reason why is because there was a mutation. So there can be no net mutations for a population to be in Hardy-Weinberg equilibrium. The next condition needed is that there's no selective mating. You're not choosing a mate. It's only random mating. So this is kind of a weird one because this doesn't typically happen, right? Random mating would be like, okay, next person that walks in the room, well, you guys are going to mate together and have offspring. So that's not very typical of human populations, right? We prefer a mate and we choose a mate based on some characteristics. It's not random. And finally, no natural selection. So the deal is you are going to have to memorize these. As I said, these conditions are needed for a population to be in equilibrium, a Hardy-Weinberg equilibrium. So you might see these as a multiple choice, or you might see these as a numerical response. You need to know these criteria, these conditions, these assumptions of the Hardy-Weinberg principle. Okay, so here's some little diagrams that show these 5 assumptions. No selection, no mutation, no migration, large population, and random mating. It's not selective mating, it's only random. As you can probably tell, we do not expect a real life population to have this Hardy-Weinberg equilibrium, right? These conditions are not realistic. Again, that's why this is a model. It's used to predict how a population may change. So if the population does not evolve, if it's not undergoing any changes in the alleles or the genotypes in the population that the people have or the organisms have, then that population, because it's not evolving, is said to be in Hardy-Weinberg equilibrium. However, we can use this model to predict that if evolution is occurring, the population is not in Hardy-Weinberg equilibrium because there's changes in the allele frequencies, the number of individuals in the population that have dominant versus recessive. Or there's changes in genotype frequencies. More recessive individuals are being born into a population, or more individuals that have a homozygous dominant genotype. So, a couple of reminders: allele versus genotype versus gene. The gene is a DNA sequence that codes for a particular protein. So, eye color protein. Maybe your DNA is ATT, GCA, TAG. That's a DNA sequence. An allele is the alternate form of the gene. You might have a dominant gene giving you like brown eyes, or a recessive allele giving you blue eyes. So just a reminder of that terminology. So in all of our examples, we're going to have a gene having only two alleles. We're not doing multiple alleles, no codominant, nothing like that, just dominant recessive. So we can use an example as squirrels. So if you've ever seen the squirrels in Kinsman Park or around town, there's basically two colors. The dominant allele, it provides the phenotype, the characteristic of black fur. The recessive allele, let's say little b for the recessive allele, that gives a squirrel a gray fur color. So we can say then that all of the squirrels that have the dominant allele, they are letter P. Letter P represents all of the individuals in a population that have the dominant allele. It's the frequency, how many in a population, and it's going to be a number between 0 and 1 that have the dominant allele. On the other hand, the letter Q is what we're going to use to represent all the individuals in a population that have recessive alleles. That's the frequency of all the individuals with recessive. So here's our formula to use for allele frequencies. P plus Q is 1. This is also found in your data booklet. So again, now we're working with two different equations, and you have to know how to use each of the equations and when. So for example, if we have the dominant allele frequency in a population, all the black colored fur squirrels, if that P value is 0.75, then we can calculate this value Q, and that value Q is for all of the squirrels in the population that have that recessive allele. So this might be the homozygous recessive, but it could also be the heterozygous squirrels that have a recessive allele. So easy, we just take this formula and we rearrange it. P plus Q is 1. So that means Q is 1 minus P. I know what P is. 1 minus P, well, P is 0.75, is 0.25. So Q, the frequency, all the squirrels in that entire population that have recessive alleles is 0.25, or we can say is 25% of the population. Typically, we do as decimal, not as a percentage, but just, really pay attention to what the question is asking and read carefully. So just a couple of reminders. We've now got two equations. We've got the allele frequencies, and we also have the genotype frequencies. So again, there's only two alleles. We've got the dominant alleles. Everybody in the population with the dominant allele, we represent as the letter P. So all of the individuals who have dominant alleles, whether they're homozygous or heterozygous, are going to have dominant alleles. The letter Q represents every individual in the population that has a recessive allele. So whether they're heterozygous or whether they're homozygous recessive, they have recessive alleles. Then the genotype equation is P squared plus 2PQ plus Q squared is equal to 1. So remember how I said I was going to talk about what each term represents? Well, the P squared represents every individual in the population who has two copies of the dominant allele. So if this was a squirrel, we would be talking about the big B squirrel. These are all the homozygous dominant squirrels. And according to our little legend up here, that means those squirrels have the black fur-colored phenotype or trait. The second term in the equation, 2pq, represents all of the heterozygous individuals in the population. So in our squirrel population, that would be all of the big B, little b squirrels. They still have black fur because you only need one dominant allele to get that dominant trait, that black fur color, to get that dominant phenotype. So these heterozygous individuals are going to have black fur. And finally, the last term in this equation, q squared, this is all of the squirrels that have two recessive alleles. These are all of the squirrels that have two little b alleles. So they are going to be homozygous recessive. And of course, we know that since you need two recessive alleles, you get the recessive trait, the recessive phenotype. So these squirrels are not black. They are going to be the gray colored squirrels because that is two copies of the recessive allele gives you the recessive genotype. And because you're the recessive, you are also the recessive phenotype. Okay, so one last time, just understanding what this genotype equation is. The p squared is all of the individuals, the frequency, the proportion in a population who are homozygous dominant. So again, those squirrels would be the big B squirrels, and of course they would be black squirrels. That term, 2pq, is the frequency, the proportion of all those squirrels or individuals in a population who are heterozygous. They have a dominant allele and a recessive. They are still going to be black squirrels. Why is that? Because for the dominant, you only need one big B to get the dominant trait or phenotype. And finally, the q squared term, this is the one that is often the most important. You're either given it or you have to figure it out in a question. Because they are homozygous recessive, let's say little b, they are going to have a different phenotype. These guys are going to be gray squirrels because they have two recessives. And that makes them easier to tell their phenotype, and therefore their phenotype is also their genotype. Okay, so there's video one. In our second video, we are going to see how to apply these formulas and do a couple of practice questions. So you might be asked to solve for an allele frequency or find the proportion of individuals that have the dominant allele frequency, or how many individuals in the population have the recessive genotype. So that is what we will do on our next video.